Hey, Carrie, remind me who's presenting today. I got Boneless Sea, H2 Pros. Who's the other one?
Sea turtles.
Sea Turtles. Okay. All right. And all right, great. I put up a, so do we know who's going first? H2 Pros?
Yeah, they asked to go first.
H2 Pros, H2 Pros, scintillating. And then who's the third one? Boneless Sea?
Yeah.
Okay, great. I will get them ready. Okay, so I've got the meeting open, and I think about 12:15, I expect to see some of the presenters joining the session.
Okay. Yeah, I was going to join in a few minutes.
Okay, no worries. All right. Yeah, if you could log in and see what you see, just to make sure I'm sharing what I think I should share.
Okay.
So, yeah.
All right, thanks.
Oh, fuck.
Hey, Dr. McDonald, how are you?
I'm fine. How are you? I'm struggling here. I need to be able to grade this. Let's see. Need to adjust the. All right. It's Vanessa.
Yes, sir.
All right, let me do. I'm trying to make sure I can grade these by teams. Let me see. Close this. Assignments. Okay, great. All right. So let's see. You want to go ahead and I'm not sure what you see. You see the announcement that class will start at 12:30?
Yes.
Okay, and I'm sorry, are you with, remind me your team, I'm a little.
Yeah, we're H2 Pros, so
Okay.
we're the first.
All right, so let's see, I can manage participants. So Vanessa, are you going to be the presenter? Who's going to be the presenter?
Yes, Vanessa Ward's going to be the presenter.
Okay, so make co-host.
Mm-hmm.
So you are now co-host, so you can share. Now, is there anybody on from the Sea Turtles or Boneless Sea?
No, it's got, yeah, it's got a little lag.
You're a little muffled. Can you get closer to the microphone? I didn't quite hear you. Zane?
Yes, sir.
Are you presenting with today? Are you with? I'm sorry, I'm not sure which group you're with, but
I'm with H2 Pros.
okay, great. Anybody from the Sea Turtles? Who's here from the Sea Turtles?
I am.
And Jackson Paddock, is that what I see?
Yes.
Yeah.
All right, I will make you co-host. And then is anybody here from Boneless Sea? Boneless Sea, Boneless Sea. All right, well, they'll check in at some point. All right, so I'm going to stop sharing the screen.
So
And
who did you appoint as the co-host for the Sea Turtles?
for the Sea Turtles, Vanessa Ward was
Okay.
co-host.
No, I am H2 Pros.
Yeah.
Okay, and then the Sea Turtles, Zane Messingale is what I see on the screen.
Also not in our group.
Yeah, I'm H2 Pros as well.
Okay, all right. Who is here from the Sea Turtles?
Me.
And sorry, I don't, tell me your name. Tell me
Isabella.
Isabella. Oh, hi, Isabella. Okay, so I should make you a co-host.
Yeah.
Okay, so you're a co-host now, and you're the co-host for Scintillating Sea Turtles. And is anybody on from the Boneless Sea? Okay, all right, so I'm going to stop sharing. Whoever is I guess that's Zane, whoever it is that's doing the H2 Pros, why don't you share your screen, which should be your presentation. So I'm going to stop sharing now, and you should have the ability to share your screen as a co-host. So go ahead and load your presentation.
All right, do you guys see it?
I do. So,
Okay.
great. All right. Now you can either do this or you can present it and do
Right.
presenter mode.
Yeah, we got presenter mode here, and I'll be using the little pointer.
Excellent. Okay. Super.
Mm-hmm.
All right. And this looks very good. And I am gonna.
All right.
Oops, H2 Pros, new rubric. All right, so what I'm going to be doing is I'm going to be filling in the abstract for your slides and presentation as we go through, and I'll be typing that into a rubric. Your final grade will depend on the extended abstract and the question set whenever that's submitted.
Right, we're not submitting our abstract until later on today because we were moved. We weren't planning on presenting today.
Okay, that's fine. That's fine. You have actually until the end of the quarter to submit all this. So if you want.
Oh, all right.
So what you're doing right now is the oral presentation, and if you look at that, that's worth 65 points out of this. So what you're doing is you're presenting the oral presentation and then the first look at the presentation slides. So that'll be in the rubric, but your final grade will not be assigned until you complete everything at the end of when everything is turned in, if that makes sense.
All right.
Okay, so okay, great. All right. So is anybody here from the Boneless Sea? Is anybody online from Boneless Sea? Boneless Sea, Boneless Sea.
Yeah.
Okay, and so I'm going to make you a co-host. Tell me who you are. I can't quite tell.
Tallulah.
Tallulah. Okay, Tallulah. Okay, Tallulah. All right, so you are now assigned as a co-host. And so that means that when we get ready to do your presentation, you'll be good to go on that. So what I'll be doing is I will start a timer, a 20-minute timer when you start your presentation. And I guess I can send you a text to let you know when you have like 5 minutes or however many minutes left, so I'm not interrupting. But do remember you need to keep on time. And so I'll be writing comments real time as we're going through this. And you'll be able to see those, and you'll be able to see the grade that I've assigned for the presentation slides and the oral presentation. So the oral presentation, once that's done, that's your final grade on that. The presentation slides, you will have a chance to revise those, but I'm going to assign a provisional grade to that. And then the extended abstracts and the question sets, those will be graded when you turn those in. In the case of the question set, that's either 20 points or no points. You know, I'm assuming that everybody's going to turn that in without difficulty, so that'll be just an either or. And that adds up to a total of 200 points for this milestone 4 grade.
All right.
Okay?
Mhm.
All right. Okay, so the presenters, I guess the first up is going to be the H2 pros. So all of the H2 pros should please unmute their mics, and then you'll be able to talk. And then just remember that you need to be aware of, you know, external sounds so that we're not picking up dogs barking and things like that. But then, you know, you'll each be able to go ahead, and you'll also be able to respond to questions from the class and from me and potentially Carrie at the end of your period. So you're going to have a up to 20 minutes for your actual oral speaking presentation, and then 5 minutes for questions, and then the next class. So it would be better if you could finish like after 18 minutes, or be really wrapping up. That way there's time for questions, and there's also time to get the next presentation lined up, and for you to go ahead and mute your microphones at that point. Clear?
That is clear.
And so we'll do that for each of the presentations in turn. So it'll be H2 pros, the sea turtles, and then the boneless sea. Okay.
I have a question. Do we have to have our video on as well, or no?
It's up to you. You know, sometimes it's easier to have your video on. People can sort of see that you're gesturing and things like that, but it's not necessary. If you just want to narrate the slides without video, you can certainly do that.
Okay, thank you.
Hey, Jack, are you here?
Yep, I'm here.
Awesome. Okay.
Okay, we only got 23 participants. I hope a lot of other people log on. I'll be stepping away for just a second.
Can you guys see my little pointer thing?
Yeah,
Okay.
it's kind of like laggy though, but either way,
Okay.
it still works.
All right. And I'll try to follow you guys with this little pointer, I guess. And like if you guys are talking about diagrams or something.
Mhm.
But that's it. I won't be using it 24/7.
Cool.
I'm just like, is it okay if I just say next slide, like when I need you to change?
Yeah, absolutely. Whenever you guys need me to, hold on. Whenever you guys need me to move.
Okay, sea turtles, remind me the title of your presentation, please.
Plastic in the ocean.
And boneless sea, the title of your presentation, please.
Anthropogenic causes of harmful algal blooms.
Okay, and we'll, as the class joins, I'll let you know this is how we plan to organize these meetings. I think this will be just fine. And I will mute my video, mute my microphone here in just a few minutes when you get ready to start. So I guess we'll, at 12:30, I'll just make a brief announcement to the class. We'll officially kick off the class. Right now, we've only got 36 participants. So, I guess I would also, you need to turn in your presentation slides. So send the PowerPoint file to me and Carrie, and then we will be putting out a notes version of your presentation, the three pages. And I know we were a little behind, but next week when we continue with presentations, we'll try to get those notes pages uploaded early so people will be able to keep notes, track along. Okay.
Do you want us to record this?
It is being recorded.
Okay.
And let's see. I think it's being recorded.
It is.
It is being recorded, yeah. So it's been recorded since the beginning, and then it'll get uploaded, and I'll upload it to the, to the regular site. Somehow the upload for the last one got uploaded to the cloud, and I haven't been able to get it out of the cloud, but I'll do that for my asphalt systems lecture. 42. Hey, thanks for going early, H2 Pros, and everybody that's going today. I really appreciate, stepping up and getting ready, being ready to go on this. That's much appreciated. And I know it's been a little bit of a scramble to get all done and get ready for this and to do it in this format. So thank you very much for, for being ready to rise to the challenge. Okay, so it's 12:30. Looks like I have 42 participants on board. I would like to have more, 43. Remember, there are going to be multiple choice questions. So everybody who is not part of the H2 Pros should have their microphone muted, except for me and Carrie. And I'll actually mute my microphone. And if I need to say something, I'll press the space bar. I'll be starting a timer. Once we get going, the H2 Pros will have 20 minutes for their oral remarks. They should aim at finishing in about 18 minutes or so. And that way there'll be 5 minutes or so for questions from class, and then time to get the next presentation uploaded and to get ready. Okay, so I've stopped my microphone. I'm going to start my timer, and I will let you know when you have 5 minutes left. I'll send you a text. Let's see, timer.
So maybe we begin.
All right, so our first presentation, welcome to class, Oceanography 4008. And of course, we have this unusual format of doing everything online. But we have 3 teams who have really risen to the challenge and have prepared question sets. So these question sets will be posted online just like the regular quizzes, and you'll respond to them just the same way. Students will respond just the same way as they do to the quizzes that I put together after lectures. You'll answer the quizzes on Canvas, and just like they are. Each presentation quiz is going to be worth 5 points. Okay, so our first presentation is by the H2 Pros team, and they're talking about anthropogenic impacts on shallow coral reef ecosystems. Take it away.
All right,
You're
so
fine.
here we are, obviously, as Dr. McDonald just said, we're talking about anthropogenic impacts on shallow coral reef ecosystems. And the 3 targeted anthropogenic impacts in this PowerPoint presentation is eutrophication, which I will be talking about, fisheries and trophics cascades, which Whitney and Jack will be talking about, and then ocean acidification, Zane will discuss. And then we have our conclusion, which we'll discuss in later in the presentation. So to begin, eutrophication is basically an overall increase in nutrients present in the water column, causing a direct increase in primary productivity. So the 2 nutrients that we're really focusing on is phosphorus and nitrogen, as they are the 2 nutrients most important in primary productivity. Nitrogen is highlighted because we will discuss that that is the limiting factor of primary productivity and is the most important nutrient input in coral reef ecosystems. Coral reefs actually thrive in oligotrophic waters, meaning there's a low presence of nutrients in the water column, and that is actually due to high levels of nutrient cycling. So you still need input for coral growth, but it's just cycled at such a fast rate that if you were to take water out of a coral reef ecosystem, the nutrient levels would be low. So the sources for nutrient input in coral reefs are upwelling and dinitrogen fixation. So with upwelling, if you actually look at where in the ocean currents and you focus on coastal areas and coastal upwelling, you will be able to see that the distribution of shallow coral reef ecosystems actually correlate with the ocean currents. Usually, obviously, the Indo-Pacific region is very well known for its coral population, which has a bunch of eddies and currents all flowing throughout that region. So dinitrogen fixation is the other form where nutrients or nitrogen is also inserted into the water column. This is done by, cyanobacteria, archaea, a lot of prokaryotic organisms that photosynthesize. So it's basically, here's like a diagram of it overall, but basically nitrogen is taken out from the air and then converted into nitrogen used for photosynthesis. This requires ATP, but it's still a very effective way to maintain and also contribute to the nitrogen levels in the marine ecosystem. If you're more interested in it, you can come back to this diagram. But overall, the main point is that it's a very important source for nitrogen. So what happens when you have an increase in nutrients concentrations? Well, actually, as I said before, corals like low nutrient levels in the water column, and they actually operate in a specific range, which this is the storage ratio, which, if you're really interested in it, you can look more into it. This paper is very, by Algalier et al., is very, very, goes in depth about the specific ranges. But basically, when you have an increase, turf algal growth takes over because coral reefs are in competition with obviously other species in this ecosystem. And usually what really happens is you have these huge algal blooms, which are actually algae in the water column, not necessarily the symbionts inside the corals, but the algae inside the water column has these huge blooms, and you basically get a loss of light, which causes coral depletion. Here are some consequences overall. Obviously, I've already talked about algal growth. Not only could there be just, an overgrowth of algae, it also could be harmful. You have toxic algae like red tide, brown tide. So that also can happen. Also overall, it changes oxygen levels in the water column. Obviously, if you have an increase in nutrients, you have an increase in primary productivity, which would lead to an increase in oxygen, but actually overall, you have a slow decrease that occurs. Here are some sources of nutrient pollution. A lot of it comes from farm runoff, like fertilizer, a lot of it's sewage, animal waste. These can all contribute to nutrient concentrations. Here are the overall effects of eutrophication, but the main one that's the most important is number 5, which is the permanent loss of biodiversity. That is the main one. These are all pretty specific, but overall biodiversity is lost. All right, Whitney.
All right, thank you, Vanessa. So I'll be introducing fisheries and trophic cascades. And so on this slide, we're just talking about how fisheries have a huge effect on the type of species that are in the ocean. So fishing down the food web causes an increase in organisms in lower trophic levels. And a trophic level is basically the position an animal has on the food chain. So yes, and you can see on this diagram that parrotfish and predator biomass right here. Next slide, please.
Mhm.
And I will also be reminding you all about trawling and what trawling is. And basically you have the long net that is dragged across the bottom of the ocean, kind of like in Finding Nemo. Yes, in that scene in Finding Nemo when Nemo like got caught in the net. But in this diagram, you see everything broken down, what is what. And an example of a species that is caught by a trawl net in the Gulf of Mexico is brown shrimp. And yes.
Yep.
All right, now
So obviously these trawling practices don't affect, or they mainly affect the fish, but when we have bottom trawling, we see that like what Whitney was talking about and showing in the diagram, the different parts of the net. When that net drags along the bottom of the ocean, and if it comes across a reef, it will basically destroy the whole reef. All the living coral and all the organisms that live there get displaced. So we can see a picture here of what a reef would look like before trawling and after trawling. And these scars that basically that the nets leave are called trawl tracks. Next slide, please. So another destructive fishing practice alongside trawling is called cyanide fishing. And it's where you basically have people like this guy in the picture diving down, trying to get aquarium fish for their aquariums. So like any sort of reef fish, like angelfish, those kind of things, those very pretty fish. But they take a cyanide solution of sodium cyanide and they spray the fish with the cyanide and it stuns the fish so it's able to be collected. But when this cyanide is left over, it actually suppresses the host respiration and the algal photosynthesis of the reef. So that can lead to coral bleaching, which eventually will lead to the destruction of the coral reef.
thank you. All right, so going more into depth with trophic cascades. So we have this diagram right here, which is a great example of what is affected negatively, and all of it is affected negatively. But you see an increase in specific species. So just look at that. And then as higher trophic levels are targeted, it creates an imbalance in food chain. And that is supported with this image. So once predators are decreasing, forage fish, they increase, which leads a huge decrease in zooplankton. And so then phytoplankton has an increase because phytoplankton was being eaten by zooplankton. And over time, the entire food chain is altered as entire trophic levels are exploited. Next slide, please.
All right, so what does this have to do with nutrients present in the water column? Well, if you can see as this little food web right here is not targeted, and you can kind of see that all of the species in the community all have this harmonized where every single species is affecting one another, and then as soon as people get in and start fishing, you have these decreases in all of these trophic levels in these species, and you're left with microalgae, seagrass, and sponges, which is on the lower side, the lower trophic levels. And when you have that, you obviously have you obviously have a change. If you have a change in species and biodiversity, you have a change in nutrient pathways, which leads to, this slide basically says that, again, I'm referring back to this paper that I talked about earlier, but fish communities act as a stabilizer. So they obviously discrete wet mass or poop, I guess, which does supply nitrogen for these photosynthetic organisms. So when you're actually fishing out these fish, you're you're taking away that nitrogen source for coral reef communities. So it's really just maintaining biodiversity, which is the biggest management plan that they have.
Ocean acidification poses another threat to coral reef environments. Corals form skeletons by excreting a fluid which exchanges ions with the seawater, forming various types of calcium carbonate. The two main forms of this calcium carbonate include calcite and aragonite. While being one of the most prevalent building blocks of corals on many reef structures, aragonite is also more susceptible to acidification due to its higher solubility. The dissolution of more carbon dioxide into the ocean results in reduced pH and reduced amounts of carbonate ions for calcifying organisms to build their skeletons. Ocean acidification is the process of increased CO2 absorption into the ocean, causing chemical reactions that reduce the pH through the formation of carbonic acid. It causes the carbonate compensation depth to rise, decreasing the range in which corals can survive. It also lowers the aragonite saturation rate, making it much more difficult for coral skeletons to obtain and keep the necessary carbonate ions. In many cases, the dissolution of calcium carbonate ions can exceed the rate of coral growth, resulting in the loss of reef habitat. The correlation of increased CO2 in the ocean bringing increases in temperature also puts direct pressure on the livelihoods of coral systems. Some anthropogenic sources of CO2 are listed on the side. Next slide, please. Ocean acidification can also wipe out many species of corals. However, some are showing promise in adapting to these changes. Some coldwater taxa have shown no major change in calcifying rates when introduced to the conditions of acidification. Scleroactinian corals have also shown increased pH in their calcifying fluid to optimize their ability to maintain skeletal growth. While these species show promise, other species are more vulnerable. This is seen with coral bleaching, or the dying off of the symbiotic coral dinoflagellates. This occurs mainly due to increased temperature in the ocean, leaving the corals to look white. The loss of the relationship leaves the carbonate skeletons exposed and without a source of energy to keep building, leaving them more vulnerable to death and algal growth. Next slide, please. This image represents the process of coral bleaching, showing the bleached white coral as a result of the loss of dinoflagellates and subsequently the overtaking of algal growth over the dead coral. Next slide, please. The impact of acidification and bleaching will alter the distribution and livelihood of many coral taxa. For instance, the corals which show increased resistance to the effects of acidification are expected to expand in size and population, while corals who are susceptible to minor changes will decrease in their prevalence. This will affect many organisms which rely on the characteristics of corals in the environment, resulting in overall changes to the physiology and interaction in the landscape. It is predicted that there will be a large decrease in the number of calcifying organisms, while the populations of photosynthetic organisms will increase. Overall, the loss of corals is expected to bring a major decline in biodiversity, altering physiological and morphological aspects of the oceans. Next slide, please. The loss of coral in ocean communities will result in considerable changes. Primary productivity and respiration rates will be affected by the increase in algal and seagrass concentrations. The growth rates of some fauna may be inhibited due to loss of habitat due to the lack of coral, while others may be increased due to higher CO2 saturation. Nonetheless, the changes that come with increased atmospheric concentrations of CO2 will alter species interactions and change the nutrient pathways which power our oceans. For example, the increased levels of CO2 affect the composition of nitrogen gas, which is used for nitrogen fixation. Changes in processes such as this will cause extreme consequences in marine life, some of which are shown in Figure 11.
Okay, so in conclusion, after talking about these three different effects on coral reefs, we can see they all have one thing in common, and that's that they're all anthropogenic. They're all caused by humans. So we come across this theory of altered baselines, which makes it very hard to study exactly where we're at, how much damage humans have actually done, because we've only started studying these coral reefs recently as our technology has advanced, but we haven't had the ability to study these ecosystems before humans actually had an impact. So we're coming into this study basically late because we don't know exactly how much of an effect we are having, and these impacts have been occurring over a long period of time. So basically the data is underestimated and the baseline of an unaltered ecosystem is unknown. Next slide, please. So all these anthropogenic factors are actually related to each other. They all stem from human expansion and our population growth. As our technology increases, so does the harm to our environment. And so basically what we can do is, personally, you can manage your carbon footprint, try to limit any sort of pollution, and also we can educate ourselves. So we included some links here to better educate yourself about coral reefs and why they're so important, because even if you don't live near a coral reef, you don't get to see them, they are very important to our oceans and the health of our environment. So thank you.
All right, and here's our literature cited. There's a lot. And we really thank you guys for listening to us. Is there any questions you guys have? All right, if there's no questions, this ends our presentation.
Thanks, guys.
Yes, thank you.
Thank you.
No, I have a question. Don't go off quite yet.
Oh, sure.
Could you talk a little bit more about turf algal overgrowth and sort of explain the difference between the different types of algae? Because you talked about how phytoplankton will bloom as a result of eutrophication, but the picture is a little bit more complicated when it comes to turf algal growth, because the grazing by parrotfish and other planktivores or other algivores is part of the picture as well. So there's like two impacts there. Could you talk about that a little bit, please?
Right, absolutely. Going back to what Whitney was talking about with the parrotfish, let's see, right here. In this diagram, parrotfish are actual grazers, which is what Dr. McDonald was talking about, which they actually target algae. They usually target algae that are on corals or more not actually present in the water column, but more actually on the top of corals. So when Dr. McDonald was saying how turf algae is different from some of the algae that is also present in these coral reef communities, basically if pelagic algae is, in my opinion, would be a more larger problem because you have loss of light. I'm pretty sure I wrote it right here. You have loss of light due to algal blooms that are located on the surface of the water column, which, as you know, the surface is where the most photosynthetic photosynthesis occurs. So when you have grazers present in ecosystems, you have a less likelihood of algal blooms because you have these fish that are maintaining and eating them. And so as you have this prey predator scenario. But with harmful algal blooms, like stated here, like red tide and brown tide, they're toxic, and not only are they toxic to humans, they're also toxic to fish, so they aren't necessarily targeted as diet. because they will obviously die if they eat these harmful algal blooms. So overall, the overall point is to maintain biodiversity in a reef, so you won't have this imbalance of photosynthetic organisms and consumers.
Okay, so there's a bunch of different effects that can result from eutrophication. One is the blooming of the single-celled algal plants, and then the other is the increase in the turf algae, which is a macroalgae. And that, parrotfish, and there are other animals such as sea urchins, which are very important browsers or grazers on algae. And so when they get wiped out, by anthropogenic impacts or other natural things, that tends to, make it more difficult for the corals to compete with algae. And so that's a part of that. All right, well, let's thank, unfortunately, we can't all applaud, but I'm sure everybody is appreciative of your presentation. We do have 60 people present, so that's great. All right, so we're gonna switch gears here. I'm gonna reset the timer.
Okay, let me stop.
And the next talk will come up. And so everybody except the members of the scintillating sea turtles should please silence their microphones, and we'll get a talk by the scintillating sea turtles. And okay, take it away, sea turtles.
All right. So hello, we're group 15, and this is Plastic in the Ocean, Increased Plastic Pollution Damages Marine Ecosystems and Health. By me, Dixie Adams. And then my other group members also would like to introduce themselves.
Hi, I'm Isabella Jaramillo.
I'm Myra Knowles.
I'm Janine Martinez.
I'm Alex Rabimbus.
And I'm Jackson Paddock.
Okay, so for our introduction, these are durable plastics that have become one of the most popular and convenient commercial items used throughout the world. From water bottles to shopping bags to medical supplies, honestly, majority of our things that we own are going to be plastic-based, or plastic in general. Plastic contents are not easily broken down, though, nor consumed by many marine organisms, making them very durable and very hard to actually dispose of. So incorrectly disposing and overusing of these items will result in a heavy polluted sea that is not easily manageable. We will discuss how both lives in and out of the water are going to be negatively impacted by this plastic in the ocean. And this is just going to give us a quick rundown of our presentation outline, and that will lead us into our first topic, which is History of Plastic in the Ocean by Jackson.
All right, so here's a timeline of the history of plastic. Here on the left side, we have the invention of plastic by an Englishman named Alexander Parkes, who first synthesized plastic in 1846 and named it after himself, called Parksine. Fast forward about 100 years to World War II, during which the production of plastic increased by about fourfold. This increase is due to the lightweight and durable characteristics of plastic that allowed it to replace other substances like glass, metal, or wood to use as a container for things. So this is where we really start to ramp up production and distribution of plastic items, and soon we'll see the consequences of that increased production. In 1965, we see potentially the first evidence of plastic in the ocean when a continuous plankton recorder, or a CPR, which is a device that is towed behind a ship and captures plankton samples for analysis, was found with a plastic bag tangled up with it off of the coast of Ireland. And a few years later, two scientists by the name of Kenyon and Cridler did a study on the birds of the Hawaiian Islands and found that 74 of 100 albatross chicks were found with plastic items in their stomach, with an average of 2 grams of plastic per bird. So this was in 1969. And then in the late 70s, we see the evidence of negative effects on marine mammals. The National Marine Mammal Laboratory, which is based in Seattle, did a study on fur seals and concluded that plastic entanglements were killing up to 40,000 seals per year. Starting in 1976, the population began to drop by about 4%, and then 30 years later, in the 2000s, the fur seal population had dropped by about 50%. So we're starting to see the negative impact on marine wildlife And then in 1997, the Great Pacific Garbage Patch was found by a man named Charles Moore while he was sailing home to his home in Los Angeles. This is a huge accumulation of debris located in the North Pacific Gyre, and it stretches from the coast of America to Japan. So after seeing these consequences, about 10 to 15 years later, countries around the world started to take action. Bangladesh became the first country to ban plastic bags in 2008, and the Netherlands became the first nation to ban microbeads in plastics in 2014, which are these tiny plastic particles that can make their way to the ocean and absorb toxins and then make their way up the food chain, which causes a lot of damage to wildlife. And finally, in 2016 and beyond, we see new technologies in development to combat this issue because it has become a big topic in environmental science. Novel tools to mitigate our negative impact, such as biodegradable polymers and plastic-eating enzymes, are currently in development. So these are some important events in plastic history. And on the next slide, there are some pictures to illustrate that history. So over here on the left, there's a picture of Alexander Parks, who first created plastic in the 1840s. And moving forward, there's some World War II propaganda about plastic Scotch tape when the production of plastic products began to increase really rapidly. And then down here, we see the effects on marine life after that increase in production. So we see the albatrosses with the plastic in their stomachs and a fur seal entangled with plastic. And then up there is the Great Pacific Garbage Patch, which is much bigger than the picture shows, but that's a pretty good representation of what it is. And lastly, there's an image of one of those new technologies mentioned before, which is the plastic-eating enzyme that scientists are working on to develop and use in the future. So we see from the beginning, when plastic is first created, to us producing mass amounts and using mass amounts of plastic, and we start to see those negative effects years later on marine life in the form of plastic garbage patch, Pacific Garbage Patch. And nowadays, we're finding ways to mitigate these negative effects that we've created. So that's a brief history of plastic in the ocean.
And so I will be presenting about the scientific principles, how plastic in the ocean relates to our class in general. So the presence of plastic in the ocean is obviously not natural. The toxins from the plastic containers absorb into the surrounding waters, which means they get absorbed by other particles or attach. And the composition of plastic is made up of inorganic chemicals that do not naturally break down, meaning the toxins and the microplastics of the broken down durable plastics are small enough to be ingested, and they don't break down. They're just there. And chemicals that are carried within durable plastics, such as chemicals with carbonate, can lead to ocean acidification. And as I was saying earlier, substances such as toxins and microplastics are small enough to be ingested and lead to harmful effects in terms of longevity and reproduction in marine life, which can lead to die-off and toxic blooms. And so all of that will then lead to physical changes and properties of the ocean. So plastic finds its way into coral reefs and shallows, destroying and altering habitats and ultimately ways of life. And this is due to ocean currents and just incorrect removal and incorrect removal of plastic and throwing them in the ocean. Invasive species are transported by plastic containers. According to one article, hard surfaces of plastics are used to transport barnacles, mollusks, and algae that are easily attached to hard surfaces into environments that they don't belong. And that can lead to the destruction of habitats. And debris, lastly, acts as an obstruction of sunlight in various environments, which we know is very important for the productivity of marine life. And starting from the smallest point in the marine food chain, phytoplankton, by them not receiving sunlight, it alters the food chain and ultimately the health of marine organisms.
And I will be extending on this topic by providing information on some Earth systems that we've discussed in class and how they impact plastic distribution and accumulation, and then some maybe other potential issues that affect marine wildlife. So we're going to look at the Earth system impacts like the ocean currents, specifically gyres in the ocean, of which there are 5. So the Earth's natural system also plays a role in how plastic debris is transported throughout the ocean. So your ocean circulation of plastic debris will bring the lightweight particles around the world. They can pile them up in various places and cause economic issues as well as issues with the habitats in that area. The birds, sea turtles, and marine mammal species are the top 3 impacted due to the plastic pollution in the ocean, which I'll touch on in the next slide in a little bit. So the ocean is unable to break down these plastics completely because of how durable they are. So they end up getting stuck for years upon like even just the micro levels. And the 5 gyres in the ocean will cause the plastic to circulate and accumulate in places for honestly until the end of time at this point, unless we have ways to combat it. And this includes 45,000 tons of plastic that is added into the ocean per year because of runoff directly or indirectly into coastal waterways. So these gyres that act as conveyor belts for plastic debris in the ocean all contribute to the global plastic load, but the North Pacific Ocean holds the highest percentage of around 33 to 35%. And this is pretty normal actually seen because majority of the total population, and specifically like the beachfront properties, they are all inside of the Northern Hemisphere. But it's also very shocking that we see a pretty high number, not as high as Northern, but in the Southern Hemisphere gyres, because there are a lot less beachfront properties and plastic contributions than the Northern Hemisphere. So these conveyor belts push around microplastic for endless years, and they progressively do get smaller as they even push them around, which then changes the movement of these particles throughout the ocean. So we can see plastic mass in the surface ocean waters dominated by large plastics like microplastics, but particle size, their count is dominated by just micros. And then really quickly, the GPGP, the Great Pacific Garbage Patch, actually includes 1.8 trillion bits of plastic, which is around the mass of 79,000 tons, and it's located in the North Pacific, spreading over 1.6 million square kilometers, which you might not know what that looks like, but imagine multiple like Spains, or even covering more than all of Alaska. And this is mostly composed of angling lines, nets, ropes, while the rest are like hard plastic sheets and films. And then the next slide will touch on marine animals, and there are 4 plausible main sinks of plastic debris. One of the bigger ones is ingestion, although there's not too much attribution of loss to ingestion. It's not only a sink that we have found, but also a large issue with aquatic life. Because these gyres that I talked about previously are international waters, this means that no responsibility is falling upon a specific country or land, which leads many independent private organizations to have to take the initiative into cleaning up the ocean by tracking garbage. So they can do this by gathering plastics in the Great Ocean Garbage Patches, finding new ways to recycle plastics, because if this is not done, marine life will continue to worsen. And as we can see, the marine life can often be tricked into ingesting this plastic, which can lead to very harmful things like hindered growth, reduced stomach capacity, internal injuries, and intestinal blockage, as well as entanglement, strangulation, reduction of feeding efficiency, and in some cases, drowning. And next slide, please.
So all of these environmental impacts also leave financial effects. Plastic pollution decreases marine ecosystem service productivity. Services such as tourism, fisheries, and transportation. It is estimated that these services offer about $49.7 trillion in annual benefits. Therefore, a 1 to 5% decline in services from plastic pollution can cost between $500 to $2,500 billion in loss. So while researchers are searching for ways to clean up some of this pollution, policymakers are doing things with hopes to slow down the amount of plastic that is getting into the waterways. There's two different avenues to do this. One is source reduction and another is green design. Source reduction includes things such as bans, fees, and taxes to stop or slow the consumption and production of plastic items. Economists often find bans to be inefficient and difficult to enforce, so instead they often recommend a slow phase out rather than immediate end. And the second avenue is green design, which includes enacting standards, extended producer responsibility, subsidies, and behavioral interventions. And these are giving financial incentives to businesses or individuals to make the switch to renewable resources or environmentally friendly alternatives.
Okay, so I'm going to talk about the societal impacts and the relevance here in Florida. So in 2008, Florida became the first state to preempt local plastic bag bans and fees. And just a few years later, they preempted most regulation of polyester, which is styrofoam. And it's worth noting that Floridians generate 32 million tons of waste every year, but only recycle 28% of that waste. Another issue that we face, and we can see it a lot in Tallahassee, is that these recycling companies and waste companies aren't finding it economically savvy of them to take recycling because they're not making as much money because it's like a heavier load, and more goes into that. So there are only like 2 or 3 student apartment complexes that have recycling. So a huge reason for this is that people don't really know how to recycle, and if you're not properly rinsing your containers and making sure certain plastics can be recycled, the entire batch is just thrown away. So on that note, I urge you to reduce, first of all, and then reuse and then recycle as a last resort. Just try not to use any plastic. There's really no reason for it. And then in 2016, Coral Gables implemented a ban on polyester, and they were sued inevitably by the Florida Retail Federation. They're pretty awful. And then there's like, you can just see today so many different environmental groups throughout Florida, like the Surfrider Foundation, Environment Florida, Oceana, and other local and statewide organizations actively fighting and lobbying these stupid preemptions. And then in 2019, Ron DeSantis actually exercised his veto power. There's a chat. He has exercised his veto power and said, you can see on the next slide right here, this is the actual letter. He said, this is just for specifically straws. He said that there was nothing wrong with individual municipalities banning plastic straws. So we still have a lot to do in terms of plastic bags and plastic bottles and cutlery, but at least we have plastic straws. And of course, municipalities are enacting their own ordinances, but they're being sued. But a lot of pro bono things are happening. Lawyers are coming in and representing them. So it's really cool.
Scientific consensus. Scientists have hypothesized that the plastic pollution in the ocean will continue to increase over the years, with recent estimates indicating that the mass of plastic released to the marine environment may reach 250 million metric tons by 2025. So scientists have also come to an agreement that there are general challenges to keep plastic out of the ocean. First, legislation doesn't get passed or activated. We see that it takes a long time for activists and environmentalists to make their point across and have plastic pollution bans. Another is the weather phenomenon keeping pollution in circulation in the ocean. A combination of the water cycle, pressure systems, and the Coriolis effect make it difficult to keep plastic out of the ocean. And ignorant and pathetic litters not disposing of trash properly can become a bigger problem. Next slide, please. Methods. So these are 3 different methods that are used to analyze plastics. The first one to the left is a picture of a manta net. They are one of the most common methods. It is used to collect microplastic, which is plastic less than 5 millimeters, mesoplastic, which is plastic from 5 millimeters to 2.5 centimeters, and then macro, which is plastic larger than 2.5 centimeters. Then we have the photographic and aerial surveys. This method is used to analyze the plastic pollution by taking photographs from the air using airplanes or helicopters. This allows for a better visual and covers larger areas. This is ideal for megaplastics. The last picture represents divers, trawl specialists, and remotely operated vehicle pilots. This method is used to analyze the plastics in the ocean floor, but it is not really used since it's very expensive and microplastic is more of a concern for marine organisms. These methods are used to examine the toxins and chemicals plastic gives off and which affects the marine ecosystem. Because of this, we have nonprofits like the Ocean Clean, which its mission is to address this issue.
All right, so here are some recommendations that we can all do to help mitigate the negative effects that plastic has on the ocean. Reduce or eliminate single-use plastic items like reusable dishes, bags, and other household items. Things like plastic water bottles, Gatorade bottles, because it all adds up and inevitably ends up in the ocean. Buying in bulk to reduce plastic packaging and recycling properly. So like putting plastic in the plastic only bin, aluminum cans in the aluminum can only bin, things like that. Volunteering at your local beach or your local waterway to clean up, and supporting policies that promote plastic bans or reducing single-use plastic items. So those are some things that we can all do, that we should all do to help combat this issue.
Sorry, I was muted. Again, in summary, plastic was produced in the 1840s, becoming popular but negatively impactful in the 1970s when the scientists from the National Laboratory of Marine Mammals concluded to be killing thousands of seals a year. The chemicals that we hear are varied, will change with every durable plastic object that enters marine habitats, and it will cause destruction in and out of the water, meaning to the species as well as the humans that live on this beautiful globe. And then humans, you can see, have been negatively impacted, as we've talked about. And then the policies being created to address the production of usage of plastic items, while scientists are using multiple methods to analyze the effects of plastic and remove plastic from the ocean.
And finally, for a quick wrap up, we noticed that marine organisms are not the only one impacted by plastic. Humans are too. This stuff costs money and it's ruining economies everywhere globally. The snowball effect of increased pollution will damage ecosystems and essentially the way of life as we know it. So I think it's important that we take note of this issue and make an effort every day to recycle, or as Bella recommended, just reuse and stop using single-use plastic. Thank you.
Okay, are there questions from the audience for this presentation?
Yeah, I have a question. How does this, I know you guys talked about like the plastic being on the coast and being in gyres and on the surface, but does this affect deep marine communities as well as, you know, coral reefs and more surface ecosystems?
Yes, it does, but pertaining to what we contributed today, I haven't been able to touch upon those lower organisms, although there is science and research being done on these deep sea marine wildlife and any type of organisms that are affected. And they are. I don't know exactly how they are, just because I did not touch upon the deep sea. I did mostly on the mammals that are up high, the birds and the sea turtles, which you wouldn't actually find at the lower levels anyway. And these are the top 3 species that are getting affected. But yes, there are, to answer the question, yes, they are. I just cannot give you for certain what they are.
I can tell you this, like we used to measure how many, when people were saying there's going to be more pieces of plastic by 2050 in the ocean than there are fish. There are so many tiny pieces of plastic that we cannot even count anymore, that the system that we're using to keep track is now parts per million. So like, and that is taking into account all those individual little pieces, these little microbes that are ending up in like all the food chains. Like if you go deep down, I'm sure you'll find like species with pieces of plastic stuck in their stomachs.
Well, thanks, guys.
Yes, I'll concur. Yeah, that they've even found plastics, in the digestive tract of organisms in the Marianas Trench. So this is an indication that's the deepest part of the ocean. So this is an indication that plastics are pervading all of the ocean. And this presentation perhaps rightfully focused on the pelagic input of plastics, but there certainly is, there are other environments that are also affected by plastics, so that the presentation did not cover, for example, coastal pollution, which is a very significant component of plastics pollution in many parts of the world and has a distinct environmental and economic impact. But the focus here was largely on the pelagic plastics. And so,
I mean, it is safe to assume that since deep sea is affected by the surface, all of the food and all the stuff that is in the deep sea usually comes from the surface. So if plastic is affecting the surface, it is obviously affecting the deep sea. Is that safe to say?
Yes, but as we know, that it takes a long time for material in the pelagic zone to make it into the deep sea. So this is kind of would be expected that you'd see these impacts and you'd see the accumulation of plastic parts in the pelagic zone and in the coastal zone before you would see that in the deep sea, but that everything is connected, so eventually it'll get there. And so that's kind of troubling. And there are plastics across the bottom of the ocean. That we encounter them with submarines and ROVs. When we drive along, we see plastic bags and balloons. Helium balloons actually turn up because they're heavier than water and they sink when all the helium is gone. So I've actually seen probably a dozen balloons at the bottom of the ocean during ROV and submarine dives. And so, the ban on helium balloons being released, by parties and things like that is also a component of plastics control. Any other questions from the audience? Okay, well, thank you very much. We'll have a round of applause at the end of this. I'll ask everybody in the class to unmute their microphone and give a round of applause for all of our speakers today. But meanwhile, we'll go on and we'll get to our final presentation, which is from the team Boneless Sea, and they will be presenting anthropogenic impacts of and causes of harmful algal blooms. So everyone else should please mute their microphones. And I guess Tallulah Belzinskov is making the presentation. And so I'll start the timer now and go ahead, Boneless Sea, take it away.
All right, so we're doing the anthropogenic causes of harmful algal blooms. So while there are harmful algal blooms in freshwater and marine ecosystems, we're going to focus on mainly marine ecosystems. So there are different types of organisms that cause harmful algal blooms. As you can see from these images on the left and right, one is red and one is green. So obviously, they are different in appearance, but also in the organisms and causes behind them. So the first one I'm going to talk about is cyanobacteria, which is a photosynthetic bacteria. It occurs in freshwater, but it also occurs in marine waters and brackish waters. Typically, a scum will form on the top of the water that's blue or green in color, but not always. They're naturally caused by excess nutrients and increased sunlight and warm water temperatures. These are areas, since they're photosynthetic, they're more productive, so they can multiply quickly. One example of a cyanobacteria that causes harmful algal blooms is microcystis, which, as you can see from this graph in the bottom right corner, it's very productive in between temperatures of around 25 to 35 degrees Celsius, where it has a lot of growth. It's the blue line. But it occurs in freshwater and produces microcystin, which is a cyclic peptide toxin that's really harmful to humans and animals. If ingested, it can lead to severe neurological effects like convulsions, respiratory failure, and death, like as you can see from those turtles in the top right. Another one are diatoms, which are photosynthetic phytoplankton. They are most common in saltwater, but they also occur in brackish waters. They can appear in a wide range of colors: reds, browns, blues. The image image in the top right corner is actually an image from NASA, and it's depicting a coccolithophore bloom in the Southern Ocean that's covering about 16% of the ocean surface. And the ratio between concentrations of nitrogen, phosphorus, and silicate in particular areas of the ocean can determine the dominance of different phytoplankton communities. So in that case, the coccolithophores, because there was a low silicate to nitrogen phosphorus ratio, the coccolithophores out-dominated the diatoms that would normally bloom in that area. And the image in the bottom right is a picture of a coccolithophore. They're also naturally caused by excess nutrients and warm water temperatures. And another example is Astroplanus carianus, which can photosynthesize in low light and low temperature, so they typically bloom in winter. And these have been found to be the cause of bleaching of Paropia, which is a genus of red seaweed that is found around the world in mainly shallow areas and intertidal zones. And that's an image of it in the bottom left. And the last one I'm going to talk about are dinoflagellates, which comes from the Greek dinos, meaning whirling, and flagellum, meaning whip. So the image in the bottom right is one. You can see it has a long flagellum, so it whips around to move. They're very similar to diatoms since they're both photosynthetic phytoplankton. They're typically found near coasts because that's where there are warm water temperatures and excess nutrients from like runoff and other coastal ecosystems. They're one of the most common harmful algal blooms, and they bloom in annual cycles typically. Some will even produce cysts so that they can survive harsh conditions, and when conditions are favorable again, they'll continue reproducing. So this is one of the reasons why they form these cyclic blooms. A big one that we've seen here in Florida is Carinia brevis, which is responsible for red tide. These two images have similar data. They're both from NOAA. The one on the left shows cells per liter and where they're highly concentrated. So as you can see along the west coast, but also the east coast, and they even extend out onto this West Florida shelf over here. And these areas where it's red is more than 1 million cells per liter, which is very highly concentrated. And yeah. And another example is Margala phinidium polycricoids. They were one of the ones found to leave resting cysts in sediment. They've been linked to a series of harmful algal blooms in India, but also along the west coast of Mexico and the northern coast of South America. They lead to massive fish mortalities because they enter through the gills and then attack the liver. And this diagram shows how when it enters through the gills, it starts to decrease liver activity, which leads to fish death.
Okay, so I'll be talking about the harmful algal blooms, and mostly they're caused by cyanobacteria, and the increase of cyanobacteria are caused by the concentration of nitrate and phosphorus levels, and mostly caused by runoff water, which can come from like the agricultural lands or like the overflow of septic tanks. Some human factors can be like the oils from the car oil, and like the runoff. So like when it reaches the land, the concentration of oil and nitrogen and phosphorus levels cause the increase of cyanobacteria and harmful algal blooms. Next slide, please. Next slide.
So Isabel, you need to click next slide, please. There you go.
Thank you. So some environmental factors are like upwelling because they're like nutrient heavy, and like those nutrients come up to the coastlines, and they kind of help increase the blooms of algae. And some factors can cause during hurricanes and like El Nino, these winds cause these upwellings to reach the coastal line. Next slide, please.
Okay, so I'm gonna kind of go into more depth about what he was saying about how climate change can affect the algal blooms. The main thing is the temperature. Um, as temperature increases and the air and and water both warm, seasonal patterns will begin to change. So a warmer spring will mean that the algal blooms begin earlier in the year, and then that also the summertime will also be longer, and so this is creating a longer window of opportunity for these harmful algal blooms to grow. And they also grow faster in warmer water. So the harmful algal blooms tend to grow faster in warmer weather and temperature, and the toxin production is sometimes directly linked to the warmer water also. The next thing is, oh, sorry, wait, go back to the other slide, please, sorry. Yeah, so precipitation also is going to affect the algal blooms. So it can be either too much rain or too little rain. Both of them will affect it. Increasing precipitation, sorry. So like storms and hurricanes can move the blooms around, and then also the increase in rain can wash more nutrients that causes the blooms to start in the first place to go into the water. And then too little rain, so like a drought, can lead to changes in the water salinity, and then so the amount of water flowing into lakes and reservoirs decreases, and then that combined with the warmer water causes more evaporation, leaving even less water, and then the salinity of the water is higher, and then when there's more salinity in the water, it's worse for the algal blooms. Yeah, okay. And then, so those are the two main things. And then, but also, so acidification of the water and stratification also play a part. Sorry. Increasing CO2 in the atmosphere leads to different pH levels in the water. And so the more carbon dioxide in marine and freshwater ecosystems favors those algal blooms, and they tend to grow faster and elevated when there's more CO2 in the water. And then these changes in the water chemistry can change the relationships between the harmful algal blooms and then the other algae that is actually good. And it changes it affects like the way that the nutrients is like available to the other algae in the water. And then stratification goes hand in hand with temperature. As the temperature of the ocean increases, it becomes more stratified, and the warming of the surface water increases the frequency, the strength, and the duration of stratification, which favors the growth of the harmful blooms. And this also affects the nutrient availability. Increased stratification leads to changing patterns of nutrient availability, and some algae can float or swim towards the top of the water, and it forms a dense surface bloom that can block the sunlight for other algae and other aquatic organisms that live in the deeper layers, and they're unable to get the nutrients that they need to grow. And then the different stratifications can lead to different hotspots in the ocean that further increases the availability of these harmful algae blooms to grow.
Okay, so some solutions for harmful algal blooms are the reduction of nutrients. So let's say, for example, choosing a type of fertilizer that doesn't have nitrite or phosphate. So when in case runoff does go into the ocean, it doesn't increase the amount of nitrogen. There's chemical technology as of right now that treat for these cyanobacteria cells. by using copper sulfate, which just kills them. And same thing with hydrogen peroxide. It is used in combination with the copper sulfate to kill these cells. And there's also technological treatment, like for example, ultrasound technology, which is used to control these harmful algal blooms. Next slide.
I'm going to just share on the effects of the harmful algal blooms, and in particular on the marine life. So the toxins that these blooms produce, they can kill the fish and shellfish directly, and they can also sicken the wildlife indirectly as organisms consume contaminated organisms or water. And in fact, more than 50% of the unusual marine mortality events are now associated with the HAVs. Exposure to toxins can occur directly through inhaling the toxins, but it more often occurs indirectly through food web transfer. And the at risk in particular are the long-lived species and highly endangered species. Mass mortalities in which a significant number of individuals perish have devastating consequences on these populations, and the repeated exposure can compromise health in more subtle ways, and this influences the species survival. And this is just a really crucial issue for our area especially, and it affects a lot of native species in Florida. In 2003, for example, 107 bottlenose dolphins were found dead, together with hundreds of fish and marine invertebrates along the Florida panhandle. And despite the absence of a concurrent bloom of the algae, the high levels of brevetoxin were measured in all stranded dolphins examined, as well as the fish prey. And this is just a really, really important study because it was the first time that brevetoxin involvement was confirmed in all the tested animals, and that the role of poisoning was unambiguously identified. This event has indicated that lethal doses of brevetoxin can be transferred through the food web, and that marine mammal mortalities can occur in the absence of a bloom even. And the picture also just shows like the humpback whales, that it's a similar, just how severe it's affecting all animals and all trophic levels. Next. Sorry, can you change the slide? There we go. There, thanks, sir. The another impact is the high biomass accumulation that's associated. Oh wait, oh sorry. Could you go back one to the sorry, can you go back a slide? Perfect. Okay, I'm sorry. The next one on the high biomass. Yeah. Thanks. This is just another effect, and it can cause light attenuation and clogging of fish gills. There is a correlation between the eutrophication and the harmful algal blooms and the hypoxia. And so this occurs when the vertical mixing is weak and absent, and oxygen depleted bottom waters are not refreshed. These conditions result in a condition of hypoxia, and it creates areas called dead zones, and they occur regularly along the northern Gulf of Mexico. And as a result of these dead zones, habitat is lost, and this puts pressure on population. And all of this leads to a decline in biodiversity. Marine life also suffers in similar conditions when blooms terminate, because bacteria breaks down the dead algae material, and this depletes oxygen from the water as well. This all leads to mechanical damage in the fish, and that could be cells lodging in gill tissues, leading to death, disruption of neurological functions, and then there's long-term effects of biotoxins on the health of aquatic animals, and this includes susceptibility to disease, suppressed immune systems, abnormal development, and even the induction of tumors. And like I said earlier, animals at all trophic levels are at risk and exposed to biotoxins in the long term, and their diet may fail, and they'll display impaired feeding and immune function, avoidance behavior, and just reduced growth. So there's a lot of important ways that the toxins harm the marine life, both physically and directly. For the next slide, sorry, you could, yeah. for the next slide, it just goes further into the food web alteration, which is a huge impact of the harmful algae blooms because it affects all trophic levels. And it's also noted that algae toxins influence ecosystems both from top down and from the bottom up. The HABs can alter food web dynamics through impacts such as physiological stress and increased susceptibility to disease and feeding impairment. And this acute or chronic exposure to their toxins, either directly or through the food web, they place certain populations at increased risk. And once again, that just is a threat to biodiversity. One particular local example that demonstrates how these changes impact the whole ecosystem's energy flow is that of the sea turtles. Harmful algae bloom outbreaks in the Gulf of Mexico occur almost annually and primarily impact the loggerhead, green, and Kemp's Ridley sea turtles. And a study showed that in 2005 and 2006, an outbreak off the west coast of Florida led to 318 documented sea turtle strandings. And while high levels of toxin in the stomach content suggested the turtles were consuming contaminated prey, it's also possible because the turtles inhale when they take their breath, they inhale the toxin. And so besides these direct effects, however, there was a lot of indirect impacts that rippled throughout the ecosystem because when the blooms are so extensive, they block sunlight and damage seagrass beds. And without seagrasses for foraging, the green sea turtles eat more algae, which may be less helpful in diet and in the long term make the turtles more susceptible to disease. And the loss of the seagrass beds affects the whole food chain with decreases in prawn and fish species, reducing prey availability for other turtle and marine species. So it just shows how it will start all the way through the ecosystem and ripple out, and the impact is really, really broad. It for my slide. Sorry, that's my slide. If who's ever next.
Okay. Are you done, Ella? I'm sorry.
Yeah, I'm done.
So I just wanted to talk about the effects of algae blooms on humans. Oh, can you go back on? Okay. So there's a huge problem if humans consume even seafood that's affected. So the most common seafood to get affected, it would be filter feeding fish and shellfish because they're directly consuming the algae. And so those should be avoided at all costs from areas that have harmful algal blooms, but also other fish, which is what Ella was explaining throughout the trophic levels, then consume other seafood. So basically, any sort of organism that runs through the system, even if they didn't die from it, are going to be harmful to humans. And there's no way to cook off the toxins. So even heat or acid, anything that you normally use to cook your food that could kill off bacteria isn't gonna like harm the toxins in any way or break down the toxins. So when they are consumed, people experience gastrointestinal problems, so severe diarrhea and vomiting, muscular aches, but then also a major problem are neurological issues. So that's associated with consumption of toxins include dizziness, memory loss, and disorientation. And they are deadly. Paralysis and death are pretty common with people who have been exposed to the toxin. Okay, so next slide, please. They also can be inhaled because the toxins can be traveled through the air, kind of like sea spray, as aerosols. So they're microscopic. You don't see them in the air. It's not like cloudy or anything. But when they're inhaled,
It can give people severe respiratory problems and cardiac problems, but also similarly to consuming them, dizziness, headaches, disorientation occur as well, and inhaling them is deadly. So anything associated with them is pretty deadly to humans. Um, and they also affect fishing industries and the economy. So, okay. Here are two examples. I picked these two because on the left is a picture of Lake Erie. Um, this one is a cyanobacteria. You can tell the bright green has covered the entire lake. And so any sort of business that's associated with recreation on the water, boating, or fishing is going to be severely affected because they have to shut down entirely. No one can really be near the water because, like I mentioned previously, it can even be inhaled. So it's shut down completely. And then the picture on the right, oh, sorry.
Hey guys, so I'm gonna run through legislation really quick. And so the federal government stepped in to help promote awareness and dedicate funding to stopping harmful algal blooms from harming marine ecosystems because we've had a lot of trouble, especially here in Florida, with like red tide and etc. Freshwater and saltwater ecosystems are looked after by the National Oceanic and Atmospheric Association and the Environmental Protection Agency. So the ones that are kind of overseeing like all of the issues on all bodies of water regarding algal blooms. And so they both monitor and protect marine ecosystems. Next slide, please. So the two like main legislations I wanted to focus on were the Harmful Algal Bloom and Hypoxia Research and Control Amendments Act of 2014. That's a mouthful, but basically, in summary, it's to reduce the damage of blooms, create policies, establish ongoing research, and protect freshwater and saltwater ecosystems. And then the Great Lakes Restoration Initiative was created to keep the Great Lakes from being overwhelmed by algal blooms because they started to see a lot of them develop. And so my picture to the right is of Lake Erie and the development of the algal blooms, which is what sparked the Great Lakes Restoration Initiative.
And that's it. Thank you. Question?
Okay, all right, so let's go ahead. We have about 2 minutes left here for questions. This presentation went a bit over time, but you did get everything presented. So the questions from the audience for the Boneless Sea. Questions? Well, I had some questions. You talked about the harmful algal blooms, and you said somewhere that the most important were cyanobacteria. But so how do you measure the relative impact of like cyanobacteria, which cause these algal blooms in lakes and so forth, with the red tides, the dinoflagellates that cause the red tides, the Carina brevis? Do you have an idea of the relative impact of these two different organisms? Hello?
So I think that it depends on how quickly they can multiply and also like how easy it is for them to grow. So like for certain cyanobacteria, they in like certain environments, like with when I was talking about the diatoms and how different ratio concentrations of like nutrients can affect how productive some are and like outcompete them. I think that might have something to do with like why certain blooms have a bigger impact or are more harmful than others.
Okay, but you don't know the relative, I don't know the answer to this either. Sort of the relative impact of all these different types of algal blooms, which one is the most, deadly to human beings or, impactful for economic effects or whatever. So that's not something that the people have investigated, or that's not well known?
No, not from what I was looking up. Mostly the research goes into specific kinds and then their specific effects, but there are definitely some that are like way less of an impact, like ones that just attack certain, algae aren't going to be as harmful as ones that, could cause death in humans.
Okay. Yeah, and you might also mention that the the shellfish toxins that are produced by some of the harmful, that's some of the most deadly toxins known to man. I mean, they're deadly in, you know, parts per million, low parts per million, even parts per billion range. And they're, you know, they immediately attack the nervous system and render people, you know, unconscious or even dead. And there have been, you know, examples of you know, fishing boats where people were eating the mussels that they caught and they got harmful algal, you know, toxins. And the whole, you know, everybody died like right away. So they found, so it, you know, that's something to consider. All right, well, I'm going to ask all of the students in the class to please unmute their microphones. And let's go ahead and give a big round of applause for everybody who stepped up first. Thank you very much. Well done stepping into the breach. And I was happy to with today's presentations. There was a little bit of overtime in the second two presentations, so that's something you have to watch for, is to make sure you're practiced and that the handoffs between slides go smoothly. You know, so that's just something to improve for the people that follow after. But okay, so there are 3 quizzes, and they'll be uploaded to the quiz section, and they'll be available for everybody to respond to. Okay, well, I'm going to stop class now. Thanks for everybody to attending, and we'll be doing this again.
I have a quick question.
Sure.
For milestone 4, is that our final version of the abstract that we have to turn in?
For milestone 4, you have you know, 4 things that I'm grading you on.
Mm-hmm.
That's the extended abstract, the presentation slides, the oral presentation, and the question set. And that will be the final grade for the team project. And you still have 200 points. So I guess the way that I set that up in the rubric, which I published, is that the extended abstract is worth 50 points. The presentation slides and the oral presentation slides, the actual oral presentation, each of those is 65 points. And then the question set is 20 points. So that's, you know, if you turn in 5 questions, you get 20 points. If you don't turn in your questions, you get 0 points. So that's a pretty easy one for me to grade. But that's, you know, the rubric has been posted and you can see that. And what I'm doing now for the folks that presented today is that I'm going to give them provisional grades for their, in the rubric for their slides and their oral presentation. And then they'll have, you know, time to submit the extended abstract and the question set. But, you know, if you can get it done right away, I strongly recommend that you do that, and that'll let you, you know, be a little bit more relaxed about the rest of the class and also getting ready for the final exam. Any other questions? Okay, well, thanks everybody for tuning in once again. And, you know, again, we have sort of extraordinary circumstances, but I'm really proud of the way you all are stepping up and getting everything done. And, you know, thanks to everyone for who presented today. And we'll be, you know, continuing with conferences. And, you know, look forward to seeing you all next Tuesday. Bye now.
Thank you. Bye.
Bye-bye.